welcome, sir.
Next is about the malignancy neoplasm conditions as such. So I'll just start the superficial issues and of course some of the latest developments also which we have to know. Now, as far as the definition of cancer is concerned or neoplasm is concerned, the oldest definition, rather established definition is tumor is a growth which is independent growth or uncontrolled growth with atypical formation and pleomorphism, and pleomorphism means mitotic figures which doesn't have any termination and it tends to metastasize. So the 5 characters which are mentioned of the cancer cells are uncontrolled or independent growth, atypical being atypical presentation, and pleomorphism or where they do not have a termination, and metastasis. Now what we mean by these 5 characters, I'll try to go into a bit of detail. Like when we say independent growth, the normal growth is considered as a dependent growth or which is under the control of many factors. Now most important that factors which control the normal growth are the normal cells respond to specific signals which actually are prompted by the body for the requirement of the cells. Or to be, or maybe if I to give a simple example, like at the time of birth, the body weight may be something around 5 to 6 kg, and maybe the height also may be 12 inches. But the rate of gain of the weight will be rapid, like the same baby would be having some 8 to 10 kgs within a few months, like, and then the same rate will continue up to maybe around age of 1 or 2 years. Then gradually that rate of gain of the weight or gain of the height also will be gradually reduced. By the age of 16 or 17, the gain in the height will be drastically reduced, and after the age of around 20, virtually there will be no gain in the height of the body. Suppose the same rate would continue, same rate as that of the child would continue, the same rate as such. So by the age of around 50 or 60, you will be sky high, but that doesn't occur because there are certain points or there are certain factors which regulate the growth of the body or which make the cells divide or control depending upon the multiple factors. And those multiple factors which control the signals or which control the growth of the body mainly are the cell cycle checkpoints. That's the DNA replication is controlled under the cell cycle, so that the number of divisions would be according to the requirement. According to the requirement in the sense like in the normal growth, at the time of the young age, the rate of growth would be more. As the age increases, the rate of growth would be lesser, and the cells also would be multiplying depending upon the requirement in the sense like suppose if there is a wound, during the healing, to fill up the gap, the cells will be multiplying at a rapid rate and the wound will be covered. But once the wound has healed, that rate again comes back to the normal and there will be no further growth in the normal situation. Of course, the abnormality would be if that growth would be continuing progress, it results in what we call as the keloid, which is a sort of neoplastic condition, benign. Maybe it's considered as a benign. So that's about one factor, one example of the growth, these controlling factors. Of course, the controlling factors are many, that controlling signals are many. At the end of the life of a cell, the cell has to be damaged, or cell either it would be dying or it would be damaged, if at all damaged due to any other issues. The cell would undergo death, a process of death, which we call as apoptosis. Apoptosis is a situation where the cell would die after the achievement of its life, and maybe the area would be completely made empty by the body, so that there is a space for the new growth to occur or new cells to occupy. Now this process also is a controlled growth phenomenon. Or there is another of the controlled growth phenomenon is what we call as the tissue adhesions or contact inhibition. Suppose if in an example, if at all you develop a tissue in a culture medium, the cells will grow at a rapid rate till the whole dish, that dish of the culture, would be having one layer of the cells. But once the cells have completed one layer, the rate of growth diminishes. There will be no further multiplication of the cells unless another layer is formed, or you just lift one layer up. So that kind of a situation is there. And to the factors which control this growth are either it's related to the genes, genetic factor, or the other is the endocrine, the hormonal situation, hormonal conditions of the body also would be affecting the rate of the growth. The other environmental factors which affect the growth or which control the growth are the nutrients and attitude of the body. Like the rate of growth in the linear axis would be more in human beings who are having a vertical orientation, whereas the rate of growth would be more horizontal in the animals which are four-legged, so on. So that's what I mean by attitude. Climate also would be affecting the rate of growth. The situation of living, the environment, either in the urbanized situation or rural situation, that also can affect the growth. Socio-economic status of the person also would be affecting the growth, and the social mobility, whether you are active or not, or the range of activity also can affect the growth of the body. Now, these rates again, they can be identified certain by the biological factors, and the identified biological factors are also called as growth factors. So, I will just summarize all these put together. What we can identify from the biological examination would be what we call as growth factors. These growth factors now are identified as again there are many of them. Among them, well-known growth factors which have a direct relationship with the neoplastic conditions are platelet-derived growth factor, or PDGF, or epidermal growth factor, or EGF, or vascular endothelial growth factor, VEGF, or basic fibroblast growth factor, or BFGF, or insulin growth factor, or IGF. These are well known, and these have a utility in identifying or maybe modifying the course of the tumor pathology to a certain extent. And in the development of the tumors, these have certain roles. Of course, a bit more detail of these growth factors we will be discussing when we discuss certain individual types of the tumors later on. That's one way of what we call as the dependent growth. Another means of what we call as the independent or dependent growth is the normal growth would be having mainly three categories of the cells, and they are called as labile, stable, or permanent. What I mean by this is there are certain of the cells which continue to multiply throughout the life in a rapid rate, like the skin cells, or mucosal epithelium of the gastrointestinal tract, or respiratory tract, urinary tract, etc. They will be continuously replenished. The rate of mitotic divisions of these cells is always quite high, rather they are considered to be the most maximum. Or hematopoietic cells, the cells of the bone marrow, or liver and spleen, particularly hematopoietic cells, they also have a high multiplication rate. Or ducts of glands also have a high multiplication rate. That once there is every day the number of divisions of bone marrow cells is considered to be around 1 lakh per day. So divisions would occur, so that you will have plenty of new RBCs or new blood cells which have a shorter life, like 20 to 21 days, and they are replenished continuously. And such cells which undergo active mitosis throughout the life of the person, they are categorized as labile cells. By nature, they are supposed to multiply rapidly. If there is that's a dependent phenomenon. Suppose if something goes wrong and this rate of multiplication of these tissues either increases beyond control, or if it goes below, then it can result in the neoplastic conditions. Another condition, another type of the cells are what we call as stable cells. Stable cells like the parenchymal cells of the organs like liver, kidney, pancreas, or the mesenchymal cells like muscle cells, fibroblasts, etc. Now, what I mean by these stable cells are, to take the example of liver. Liver grows rapidly at the pediatric age, but once it achieves a moderate size, the average size, then there will be no multiplication. But if at all, suppose you remove a portion of the liver, as in case of the liver transplantation, a donor liver, a portion of the liver may be removed. Now, that removed tissue would be filled up rapidly within a short duration because there is a demand for the tissue. And once there is a demand for that function, the rate of multiplication increases, but it increases only till that level or the capacity is filled up or requirement is filled up. Further, there will be no more increase in the size of the liver. Or similarly, as in case of any other tissue, as I told about the healing tissue, any healing tissue, anywhere, muscles or maybe the bone or anywhere, once there is a healing process, the healing process will continue till that stage where the gap is filled up or the average tissue pressure is maintained or the function of the tissue is regained. But once that is achieved, the cell growth again comes back to the original resting condition, and such cells are called as stable cells. Now, suppose if there is a continuous stimulation, uncontrolled stimulation to multiply for these tissues, they also can be a neoplastic condition. Example is the cirrhosis of the liver. A cirrhosis of the liver starts with the necrosis of the tissues in the liver due to any other pathology, and that necrosis may result in a continuous stimulation of the tissues to develop, and this continuation may progress into a situation where it can become malignant, or as in case of some non-healing ulcers, it is also called as Marjolin's ulcers, a ulcer which doesn't heal properly and the healing process has become abnormal. There will be a continuous stimulation for the tissues to grow, and at one stage it may become malignant. So on. So such is again the example of what we mean by the dependent growth converting into independent growth. Another type of the cells are what we call as the permanent cells, which do not multiply, like the nerve cells, cardiac muscle cells, the skeletal muscle cells, lens of the eye. The number of cells which we have at the time of the birth they are the same. They would not increase in number, but their size may increase. Cardiac muscles or skeletal muscles size may increase, the number remains the same, and it doesn't really multiply to a great extent. Of course, skeletal and cardiac muscles, they can multiply to a certain extent. Nerve cells and lens of the eye cells do not multiply as such. Of course, some new research says that even the nerve cells do multiply, but anyway, in general, they do not multiply in the average life as such, and that is considered as the permanent cells. Suppose these cells lose that kind of a character of being stable and they start multiplying, that also becomes a neoplastic condition. Now, another process of what we call as the dependent or independent growth is the process of differentiation. Now, this process is differentiation in the sense, at the time of the birth, the whole body starts from a single cell, which we call as the totipotent cell or embryonic cell. That embryonic cell has a capacity to produce all forms of the tissues which are in the adult body, starting from the skin to the nerves. So it has that potency, and hence such cells are called as totipotent cells. As the cell division occurs, in gastrula, morula, and so on, once at one stage we consider the embryonic growth of the three phases: endodermal, mesodermal, and ectodermal phases. Now, endodermal tissue can produce only the mucosa of the gastrointestinal tract or maybe the urinary tract and so on. Whereas the mesodermal tissue can produce these muscles, bones, and so on. Ectodermal tissue produces either the skin cells or the nerve cells. That the capacity of these cells would be reduced from the totipotent. See, endodermal, ectodermal tissue, if at all it is allowed to multiply, it can either grow into epithelial tissue or nerve tissue. It cannot grow into a muscle tissue. Whereas a embryonic cell, It can grow into any type of the tissue, and that's what we call as a pluripotent cells. These cells are having multiple capacities. So multipotent cell is the next in the generation. Then you have the oligopotent or unipotent cells. That oligopotent or unipotent cells are what we call as the blast cells or the adult cells, and adult cells are like a tissue, epithelial tissue from an adult body. If at all you allow it to multiply in a medium, it grows only into epithelial tissue, doesn't grow into the nerve tissue, which otherwise would have occurred in ectodermal tissue. So that kind is called as unipotent cells. So the whole growth of the body would undergo a process where the totipotent cells becomes a pluripotent, and then it becomes multipotent in between, and then it becomes a unipotent cell, which is seen in the adult body or fully formed body, and this process is called as differentiation. So there is a gradual progression in the gaining of the functions, but at the same time modification of the structures to achieve those functions, and this process is called as a differentiation. And a normal growth always follows the path of differentiation, where it starts from the totipotent cell and then the gastric morula or so on, and then it becomes the unipotent cell as such. That's, whereas if at all this pattern is altered, and if it goes in the other way, like instead of the epithelial tissue, as it multiplies, it tends to resemble that it's a precursor cells, like either nerve cells or maybe the other precursor cells, earlier forms of the cells. That process is called as de-differentiation. Now this also is a sort of independent pattern, and this independency also is suggestive of a tumor pathology. So that's what we mean by the dependent or independent growth. I have given a few examples and explained that whole phenomena. The other is the atypical arrangement in a tumor tissue. If you examine a tumor tissue under the microscope, the arrangement of the tissues would be something different from the normal. The normal tissues would have a typical arrangement, like skin epithelial tissues would have a flat orientation where one layer would be the other. The skeletal muscle tissue would be arranged parallel to each other, and that's exactly what we make out when we identify the tissues under the microscope. Whenever you examine the tissues under the microscope, identification of the tissue is based upon its shape and orientation. Now, in a normal pattern of the tissue, every tissue would have its own pattern, like as I said, muscles, there would be parallel orientation of the cells, skin, a flat orientation of the muscles, cells as such, liver, you will have that typical lobular structure, the central tube, and then the tissues around in the form of sinusoids, or so on and so forth. So every tissue would have that character where the tissues are oriented. Now suppose this orientation is lost and the tissues have a orientation which is not normal. That's what is the initial sign of a tumor pathology. So whenever we diagnose a tumor, the primary confirmation of the diagnosis would be by histopathological examination. Examination under the microscope is the most dependable method of diagnosis. And what the pathologist would do under the microscope would be, one is identifying the tissue and whether the tissues are oriented or not. If the normal tissue just changes its polarity or direction, like when I say polarity, it means every cell will have a property of direction. Like, or simply to give an example, if there is a glandular epithelium, the glandular epithelium, the secretory face will be towards the lumen, whereas the non-secretory face will be towards the opposite side. If the arrangement is the other way, instead of being at that kind of orientation, if the arrangement changes, then it's a sign of a neoplastic growth, so or a tumor pathology. So these orientations or disorientation, atypical, may be presented as either a change in the polarity, or it could be seen as the geometric configuration. Like if some areas would show a extreme high curvature or a low curvature, or If this tissue crosses the basement membrane, if a cell crosses the basement membrane, so every tissue would have a plane which decides its limits. So if a tissue crosses the basement membrane and goes beyond that, then it may be a sign of a neoplasm. So a pathologist would be identifying these characters under the microscope, and that's why whenever you send a tissue for histopathology, it's necessary that you have to send tissue from the expected border of the tumor, so that the pathologist can identify the normal orientation of the tissue compared to the pathological changes which have occurred, and hence the diagnosis would be possible. So atypical arrangement is something like a haphazard arrangement in the classroom. When I say independent, the physiology is a dependency. Independency is a sign of malignancy. So when we say independent day, it's that you have a tendency for the neoplasm, and when the alignment or the organization of the group of people is not proper, then also is suggestive of a tumor pathology. Now the same atypical arrangement also could be seen as either a increased density of the tissues, what we call as a jamming, where in a normal orientation of the tissues, the size of the tissues would be, and the size of the cells would be uniform, and the gap between the cells would be some pattern like. If suppose in some areas there is increased number of cells, that's what we call as jamming, the same common word, and this also is one of the features which is seen in under the microscope. Or if it is jamming, either it could be at the same plane, or jamming could be making the cells shift to the other planes, having that multiple planes, which results in increased interstitial fluid pressure and after the constriction of the cells, and hence the shape of the cells may become abnormal. That also may be the sign of the atypical arrangement. Now, as a result of this collective invasion, that the tissues becoming higher density, there is a tendency for the tissues to get broken down, and the cells may just move into the circulation. So that circulating tumor cells would be formed, and if at all identification of a circulating tumor cell is done, that's a tumor cell from anywhere may get into the circulating fluid, either the lymph or the blood. That also is a atypical formation. Now another issue is that dedifferentiation or having the mitotic figure. Now what I mean by it's one of the important criteria in making the diagnosis of the tumor pathology. A tissue or a cell which undergoes mitosis would have the typical changes seen in the nucleus, and the nucleus would have, and as you have studied in biology, these phases are considered as prophase, metaphase, anaphase and telophase, the normal. In the prophase, the nucleus will become bigger, having high staining property. Gradually, the metaphase, the cells become the nucleus becomes elongated and shows the evidence that there is an increased nuclear material. And in the anaphase, there will be polarity, that the nucleus tends to get separated into two separate compartments. Telophase, even the division of the cell, the cytoplasm also would show the evidence of that cleavage, and it forms the independent cells. In a normal tissue, adult normal tissue, when you examine under the microscope, particularly in case of the stable cells like the skin or the liver, the number of cells which will be undergoing these phases, any phases of the prophase, would be less than 10 per 100 cells counted. So under the microscope, when you count 100 cells, the number of cells which are undergoing that active mitotic division, which may be present in any phase, prophase or the telophase phase, would be less than 10 per 100, that's less than 10%, or usually around 4 to 5%. But in case of a tumor pathology, the number of such cells would be more, and that's what we call as a mitotic figure. And these mitotic figures are atypical. They, again, the nucleus also, the shape of the nucleus also could be altered. Instead of that typical pattern of the normal pattern of the mitotic phases, where the cells would be, the nucleus initially would be enlarged in size, then becomes elongated, then there is a cleavage. Instead of that, the shape could be altered. Either it could be triangular or maybe crossed as such, or multiple legged or flattened, and so on. Now these patterns, now they are named with that different nomenclatures, A to H, the typical patterns which could occur, which and the number of cells under the mitotic phases also could be more, and this is what we call as a mitotic figures. And this number of mitotic figures would be a sign of malignancy, and it also gives you an idea of rate of the growth of the malignancy. A growth or a neoplastic growth which has a high mitotic figure would be more malignant, whereas 10 to 14 mitotic figures, when I say 10 to 14, the number of divisions seen under the 100 among the 100 cells which are counted under the microscope, if the cells which are having mitotic figure in between 10 to 14, they are considered as benign. Less than 10 is considered as normal. If it is more than 14, it is considered as a malignant, and number being higher, higher the number, the possible pattern, this malignancy or rate of growth of the tumor also would be faster, and that also gives you an idea like the severity of the tumor as well as the possible prediction of the susceptibility of that growth to your treatment protocols like radiation or chemotherapy. Moreover, the abnormal mitotic figures, that abnormal shapes of the nuclei seen in the mitotic phases, also would suggest about the severity of the malignancy. So now again, this all my purpose of showing this is to give you an idea of the complexity of the diagnosis of a tumor. Ultimately, the diagnosis depends upon what the pathologist would observe under the microscope. So, suppose if the pathologist is not having that patience of seeing these cells and the patterns, or misses these patterns, then the diagnosis could be negative. Or if he is over enthusiastic and maybe identifies these wrongly, there could be an abnormal or overreactive diagnosis as such. These atypical mitotic figures are one of the important criteria which make the diagnosis and which help in the diagnosis. And see, this differentiation between the normal mitotic figures and atypical mitotic figures, they are very minute, and these minute differences have to be noted by the pathologist. If the pathologist misses these, then the diagnosis will be abnormal, or a diagnosis will not be dependable. Now this is again shows the complexity of the treating or diagnosing a neoplastic condition. Ultimately, the final diagnosis would be based upon my histopathologist observation, and histopathologists may miss these due to many issues. Either maybe the cells which you have sent for the histopathological examination, the biopsy products, may not be sufficient. The cells might not be in a proper condition. They may not be stored or maybe transported in a proper condition, or the staining technique may have some errors, microscope may have error. Ultimately, the observation of the pathologist may not be capable of identifying these. So the scope for error in the diagnosis of a tumor is quite high, starting from the biopsy to the final pathologist opinion. And unless every protocol is followed properly, the diagnosis of a tumor becomes very difficult. Now, to make it fine-tuned, one of the recent developments now in case of this microscopic examination is the immunohistochemistry. Immunohistochemistry is just introduced into practice very recently, something around 20 years only. Earlier, immunohistochemistry was not known. It was only the staining, usual hematoxylin eosin stain was used. Now they use immunohistochemistry, where the immune bodies are used to modify or identify the shapes of these cells and to assess the mitotic activity as well as the binding of the cells to the antibodies, so that the diagnosis would be more accurate. Now this immunohistochemistry itself is a very complex branch, where the type of the antibodies to be used to stain the cells should be very case specific. are type specific. So it's not that there is a universal that immune chemical which can be used for all tumors. See, as far as a hematoxylin-inducing stain, irrespective of the type of the tissue, H&E stain is used and seen under the microscope. But with the immunohistochemistry, the immune staining substances or immunological staining substances could be specific, and the whole process is complex. Every simple laboratory cannot do this, and but still this is considered as one of the major advances in the diagnosis of the tumors, by which the accuracy of the diagnosis is enhanced. So ultimately, the diagnosis is subjective. Diagnosis has to be based upon the observation of a pathologist, but the accuracy can be enhanced with these recent techniques. The immunohistochemistry is one of that the recent developments. Now, anyway, this is about the other character, the atypical arrangement and the pleomorphism of the cells, like in multiple shapes of the cells is the character. Now, one more character of the tumor which I have said is there is no termination. When I say the termination, a normal cell, once it has its achieved life, it gets it dies on itself. The enzymes which are responsible for the death of the cells, they are cytolysins, they are already present in the cell. At the time of the birth, the formation of the cell itself, the total duration of that life cycle of that cell is predetermined, and once that is achieved, the cell undergoes lysis by a process which we call as apoptosis. The normal apoptosis would be forming with the gradual process. Initially the cell shape would be altered, nucleus gets shrunken, then the nucleus gets broken into smaller pieces, and then all the cell component would be breaking into multiple small breaks, and then they become completely dissolved. This is what we call as a normal termination. In a normal termination, the whole process undergoes that stage by stage, whereas a cancer cell either doesn't go apoptosis, and even after its lifetime, it continues to divide. The nuclear material goes haphazard, the shape also becomes haphazard. Now, these, of course, when I say the tumor cells do not have a termination, that doesn't mean that the tumor cells would never die. Tumor cells also would be dying, but even that apoptosis of the tumor cell, if they undergo that apoptosis process also is not normal. Instead of having a complete lysis in a normal apoptosis, the cell would undergo that different phases, and then the whole substance becomes liquefied, so that there will be no further or there will be some space for the new tissues to grow, and all that evidence would be some apoptotic bodies may be seen. Whereas in case of a tumor cell, even the apoptosis, even at the end of this apoptosis process, the apoptosis bodies would remain, and they also can give extra pressure to the cells as such. So that's how a apoptosis process, a cancer cell may die. When I say no termination, I don't mean to say that the cancer cells would be continuous or infinite life, but some of the cells may die, whereas by the time the other cells will be multiplying at a rapid rate, and the rate of growth of the tissue would be much more than the normal. Or a cancer cell may even undergo necrosis. There could be a necrosis also, and even at that end of necrosis, there will not be a lysis. The cell content may again present over the area. Then autophagy, that's a destruction by itself, which doesn't occur in case of the cancer cells, whereas autophagy can occur in the normal cells. So now, when you look at the microscope about the tissues, the evidence of these apoptotic cells or absence of apoptotic cells or these abnormal patterns also can give you some idea about the possible tumor pathology. So ultimately, the diagnosis of a tumor, identification of a tumor is based upon the histopathological observation, the observation seen under the microscope, and to summarize those factors which are seen under the microscope to make the diagnosis would be the in a typical pattern, the shape of the cells would be having normal cells would have a regular shape depending upon the type of the tissue which you have studied, whereas the cancer cells would have irregular and their shape would be altered, size also would be different. Growth and cell division in case of the normal cells will be controlled, whereas in tumor cells will be uncontrolled. And what I mean by that, normal and abnormal, we have discussed already. Apoptosis in case of normal cells is present, whereas in the cancer cells it's either absent or having that abnormal process. Communication with the nearby cells, like some substances exchanged between the normal cells nearby would be in a normal pattern in the case of normal cell, whereas in the cancer cell it's not present over there. A normal cell nucleus would have its own pattern, usually compared to the whole size of the cell it will be smaller, or of course depending upon the type of the cell the ratio may increase. The capacity of the nucleus to stain would be having a typical pattern compared to the light stained, whereas in case of the tumor cells the nucleus would be larger and would be having a darker stain. New blood vessel formation around the tissue would be seen in case of normal cell at the time of the initial growth, but once the tissue has grown, has reached the maturity, there will be no new blood vessel formation, or angiogenesis is not seen. Whereas in case of cancer cells, there is a continuous process of angiogenesis, that's new blood vessels are formed around the growth. So when I say, like when we collect the tumor biopsy from the edge of the tumor, one of the observation at the edge is that new blood vessels seen under the around the area, which again gives you an idea about whether the tumor is neoplastic or otherwise, the tissue scent is neoplastic or otherwise. So another of the observation a pathologist has to do. The process of repair by either the primary healing or the secondary healing seen in normal tissue if there is an injury, and that process of repair also has its own microscopic pattern, like fibroblasts growing into the fibrocytes acid, whereas in case of the tumor pathology this process of the tumor is absent. Of course, spread of the tumor, metastasis, I will be coming to that part, like what we mean by metastasis and how it occurs and so on, bit more detail, is present in the is not present in the normal cells, whereas in the abnormal cancer cells it will be. A glycolysis in case of normal cells is always by aerobic, that by using oxygen, whereas the tumor cells it can result in abnormal glycolysis, that breakdown of the glucose into carbon dioxide and water doesn't occur completely, and that results in accumulation of the acidic substances. These acidic substances, lactic acid or other acidic substances seen in the tumor tissue, also can be one of the means by which a diagnosis of a tumor is possible. So these pathologists may even study this acidic content, like in case of the staining process, the acidic stain would be alkaline stain would be more taken up by the tumor tissue, because it has a more acidic substance, whereas the normal tissue would have a balanced take up of the acidic and alkaline stain. Then DNA synthesis or RNA synthesis in the tissue, in the nucleus, also is one of the character, whereas in case of tumor cells, the RNA DNA synthesis will be more. That gives you the abnormality in the shape of the nuclei. The capacity of the nuclei to take up the stain also would be higher. Then catabolism of pyrimidine. Pyrimidine also is one of the essential substance which gives a staining character to the cells, and this also can give you some idea. And the tumor suppressor genes are present in the normal cells, whereas now this is one of the latest observations under the electron microscope. The gene shape could be identified, and that tumor suppression gene TSG can be identified under the electron microscope, whereas in tumor cells these are absent. The contact inhibition, that's pressure between the cells, would be normal in case of the normal cells, whereas in tumor cells it will be abnormal. Now, summary of these, all of these or maybe a few of these characters are observed under the microscope to make the diagnosis. Now the next part which we discuss is about the tumor metastasis. Now, or how the tumor occurs, the whole process of a tumor occurring, what makes the tumors to grow, the causative factors. There are a lot of theories about how the tumors are formed, whether the tumors are formed instantaneously or it's a gradual process. This is the controversial issue. Now, at present, the current theory is a tumor cell starts from a single cell. Say, one of the cells in the normal pattern would have escaped the normal rules of the growth and it starts multiplying. In a normal phenomena, there would be always, when the tissues are multiplying in the body, there is always an accidental chance that some of the cells may just miss the normal pattern and they may become abnormal. But in the normal situation, there is a scavenging system in the body which destroys these abnormal cells divisions, and one of them is called as a tumor necrotic factor, or TNF, tumor necrotic factor. It destroys those cells which may have that abnormal orientation or abnormal formation during the growth. But if somehow these abnormal cells miss that scavenging system, they tend to multiply. And this, once they start multiplying, there will be mutation. The genes would continue to change. The pattern of genes would continue to change. And to have an abnormal cancer cell, now it is expected that at least there will be 6 phases of that mutation of the genes, which requires around maybe 2000 multiplications. One cell multiplying into 2, one mitotic division. If this has a abnormal tendency, one of the products of that mitotic division has abnormal tendency, still it has a high possibility that it may be, that abnormal cell may be destroyed by the scavenging system of the body. But suppose if it escapes, then the genes in that cell would be again further modification, and this modification is a gradual step-by-step process. And as per the current concepts, theoretical concepts, it has to undergo at least 6 phases of the mutation. The genes have to modify it at 6 stages till it becomes an independent cancer cell. So, all I mean to say is that the formation of the cancer or a tumor pathology in the body is prevented by the normal process of the body, normal physiological phenomena of the body very effectively in the normal condition. But if somehow these effective mechanisms are not capable of doing this, then it may become a tumor pathology. And certain of the basic issues are, the normal cell needs to undergo at least 6 mutations before it becomes a cancer cell. And one tumor, identified clinical tumor, would have at least 1 billion cells. So by the time a cancer is formed, the number of divisions which has occurred would be quite huge. And cancer starts from a single abnormal cell that undergoes changes which can be inherited into it. Now this gives an idea of 2 aspects. One, the formation of a tumor or a generation of a tumor pathology, a cancerous disease in a human body, has many factors which can be prevented naturally, physiologically. There is a mechanism of prevention. The other aspect is when we say early diagnosis of a tumor, the early diagnosis of a tumor is virtually not possible. Identification of a tumor division, that abnormal division at the first stage, is still not possible, because by the time you make the diagnosis of a tumor, it will always late, at least there would be 1 billion cells or at least 1 billion divisions. Now certain of the theories about that, how this whole process starts, would be the oldest of the theory is a hypothesis that cancer originates from a small population of embryonic cells, is the oldest theory, with the embryonal rest theory of Cohnheim, which is one which was considered to be very old and still it is existing. Some of the totipotent cells may be remaining over there, and they may multiply later on due to some activation. They remain dormant. Of course, in case of dermites, this is true. With the dermites, this is true. Like a tissue, a cell of some other active phase, maybe the pluripotent phase, may remain dormant and later on it may again multiply. So, in case of the dermites, like ovarian dermites, you will see that dermite growth with maybe hairs, bones, etc. in the ovaries. That's due to some mesodermal cells which were remaining there for as dormant, and later on when they get stimulus, they may multiply and they may produce what we call as dermites. Or the other theories are somatic mutation theory, which says that there is a gradual mutation of the genes of the tissues, and these genes may be altered into oncogenes. The tumor suppressor genes which are supposed to be present in the genes, either they become dysfunctional or they may be absent, and hence the tissue tends to undergo a mutation. This is another of the theory. The other theory is the tissue organization field theory, which considers the proliferation of a default state of all cells in carcinogenesis as a tissue organization abnormality. Tissue organization pressure, like the examples would be due to continuous pressure or irritation of the cells. One of the usual examples given is chimney cancer. Chimney cancer is a occupational disease where a person who cleans that chimneys, due to exposure to the carbon and that irritation, the physical irritation of the carbon, the tissues become disorganized, and that disorganization of the cell itself can result in the neoplastic condition. That's the basis of that tissue organization field theory. Or another of the very rare type of the tumor also is called as dhoti cancer. Dhoti cancer is in the southern India, people tie the dhoti around, and due to that continuous compression of the dhoti around the buttocks, around the abdominal wall, the tissue may become carcinogenic. So this is about the theory and example about that. Then there is a atavistic theory of cancer, or ATC, which postulates that the capacities of cancer pre-existing in a human cell, every human cell has a tendency to have a cancer growth, and if these cells become reversed, that process of the reversion of the ancient program, which is triggered by again mutations, the older characters may be again reactivated and it may become a tumor pathology. So this is about the tumors which are transmitted through the genes, many of the genetic conditions. When we discuss the individual tumors, I will give some examples of this atavistic theory also. Then the cancer stem theory is another of the theory, where the tumor initiating cells are present like a stem cell in the body, which may renew later on, and this is seen in multiple types of the tumors in a patient occurring. Now, genetics in the cancer is one of the important developing areas. The most important genes which are now identified and which are considered to be responsible or having an effect on the growth of the cancer are oncogenes or tumor suppressor genes or DNA repair genes. Now, these are in the focus, and they also have been used in the treatment of a tumor. Certain of the oncogenes which are well identified and which are still used in the current clinical practice are related to the breast, where HER2 genes are identified. Esophagus, colon and pancreas cyclin genes are now used as such in both diagnosis as well as therapy. Pancreas, KRAS genes are 4 varieties. Colon, the catenin genes. Liver, cyclin genes are identified. Melanomas, mutant genes are now identified. These are certain genes which are used in the oncogenetic issues. Then comes the other issue like carcinogens, the factors which induce the cancer disease. We will continue to discuss in the next session, and we will wind up today. If there are any questions, okay, so we will wind up today.